While our next speaker sets up a laptop quickly, I'm just going to introduce her. Prof. Karin is a mycologist that specializes in the taxonomic of Ascomycetes fungi and the role that these organisms play in the environment. Fungi are vital in the recycling of carbon, nitrogen, and phosphorus in the soil. Very little is known about the species that are present in our soils and the relationship with plants, and our research group are looking into it. We're also looking forward to working with her next year on the new trials we're starting, and we're looking forward to listening to her. Thanks, Karin.
Ja, dit is altijd een gamble. Gaan het werken of gaan het niet werken? Het werkt nog niet. Werkt het nou? Wel, ik sê altyd vir my voorgraadse klap as nie werk nie, druk knoppies tot iets gebeur. Wat sê? Nee, is right. Ek sê nou maar, oh, there we go. Of PowerPoint het 'n probleem. So, terwyl PowerPoint besluit om op te start hierso. Miskien net vinnig voorstels by Jammer Johanne daai van die web afgetrek. Dit is eintlik 'n bietjie van 'n ou een al. Ek gaan maar in Afrikaans 'n bietjie praat en dan later in Engels. As sy, baie dankie. Ek is 'n klassieke microbioloog opgelei in swamtaxonomie, wat party mense sê baie boring is. Ek vind dit geweldig interessant. En as hierdie nou weer bom, dan gaan ek huis toe. En my belangstelling in grondmikrobiologie kom van my postdoc wat ek in Canada gedoen het. I had an exciting postdoctoral supervisor who said, "What would you like to do?" I said, "I don't know." He said, "Go dig in the garden." So I did my postdoc at an agricultural farm, so there was a lot of things in that garden to find. En dit is waar my belangstelling vandaan gekom het. So my interest is invasion biology. I'm interested to see what happens if you disturb soils, what happens to the microorganisms there? It turns out the question is a lot more complicated than I anticipated, so I've learned a lot. What I will tell you is that I cannot tell you what to do, but maybe just make you aware of technologies that are currently available to test soil microorganisms and to look at them and to do a lot of fun things. And if you want to spend money, I would be more than willing to help you. Okay, so usually people ask you hoekom wil hy nou nie aangaan nie? Moet jy hom laat aangaan? Okay. Goed op dit. There we go. So people would ask you, what is a healthy soil? Well, it all depends. It depends on what you like to do there. Would you like to farm? Then you think that is a healthy soil. If you like a forest, that would be a healthy soil. Or maybe something like that if you want to grow eucalypt trees like we do in South Africa. I don't know if you've seen it, we grow it like wheat fields. So they're all in there. But if from a microbiology standpoint, these are deserts. They're very, very low in diversity. They only have a couple of things in there. But maybe, like me, you're interested in fynbos. So I'm very interested in the fynbos. I like to know what happens if we replace our fynbos with invasive acacias and eucalypts, or sometimes what we do if we do agriculture on fynbos soils. Maybe this is a healthy soil. So it all depends on what your question is. So we define it as the capacity of the soil to function as a vital living ecosystem that sustains plants, animals, and humans. Now, I don't have to tell you that soil is probably one of the most complicated biomaterials on the planet. You have a variety of things in there. We have fungi that keeps everything together. We have bacteria. We have protozoa. There's worms. There's all kinds of things in there. But for this talk, I'm just going to focus on the microorganisms. So if you take 1 gram of soil, potentially you can get up to 10 to the power 11 different bacterial cells in there. That's a lot of bacteria. Now, I don't know how many of you follow what happens with the human microbiology these days, but we now know what happens on our skins and on our gastrointestinal tracts. And as you sit here, each of you have your own unique set of we call it a microbiome. So if we take a sample from you, next week I can identify you just by looking at your microbiology. I can know you were there. So that's very exciting. And these are very self-sustaining ecosystems. They would, and people are very interested in this. And we know that once we disturb these things, it's hard to get them back on track. So if you want to think about soil as a living organism, we can say it's very similar to the human microbiome, which we're very interested in these days. So soils have a number of ecological functions, decomposition, and I'm not going to read through all these functions, you know all of this. But many of the functions of our bacteria and fungi are very unclear. So people usually want to, when they come and see me in my office with a little bottle or a little baggie of things, want to know what's in there. Well, I don't know. It's very hard to find out. What we do know is it's not a homogeneous mass, and this is something that ecologists are still debating. You know, does everything occur everywhere? You know, if I take a sample from the Swartland, it will be very similar. The similar microorganisms found on Table Mountain. The answer is no. Everything does not occur everywhere, but we can't explain why. So we're still working on that. What we do know is if your soil diversity, and with diversity we mean microorganisms, are high, your soils are healthy, because they do all the things they're supposed to do. If it goes down, soil health goes down. And we do know if we practice agriculture, soil diversity goes down, and with that, soil health goes down. But you can do things to make it better. Well, I thought the first thing that I'll share with you is how do we measure them? So it's very easy to say as how many are there, and that's a different question. So we measure it in different ways. Many of you have been through a microbiology lab, maybe, you know, we put them on plates, we count the colonies, we do a plate count. That is probably the most inaccurate way to do that you can possibly think about, unless you're looking for something very specific. They have very specific growth requirements. Some of them need to grow with their friends, otherwise you won't ever see them. Some of them will never grow on a plate. So we can culture less than 1% of microorganisms in soil. So we've put them on a plate and we try and count them. We're only counting 1 to 2% of microorganisms. And those excludes the ones that may be important in the function that you're looking for. But it's very inexpensive, and that's why people do this. Some of the other things we can try and do is we just take a soil sample, we take out all the DNA in there, so everything has DNA, and we use that as a fingerprint. Now, that is quite useful, because you can do a lot of things. Once you have the DNA, you can do a lot of things, and we can ask a lot of questions, and I'll show you some examples of what we've done so far. These are just some of the techniques that we can use. So let me start off, and I thought, let me start off with a case study. We worked with the ARC at Blaauwklippen looking at cover crops in vineyards. We work with vineyards, there's a lot of them around. So looking at the effect or the impact that a cover crop would have on the microbial community. So the hypothesis was if you put it there, the diversity would increase. Now on the top row is the diversity indices, in other words, how many species occur there in a given season. So these are over 3 years, and as you can see, well, it didn't matter. No impact whatsoever. The diversity did not increase with the addition of cover crops, and we had a variety of treatments on these plots. And then we thought, well, maybe there's a difference between the work row and the bench row. So because obviously they would be different. Turns out there's no difference in the diversity. So same numbers, which brings us back to when you want to do these studies and you read the papers, read carefully what they measure. Because what this does not tell you is who is there. It tells you how many of them are there, but it doesn't tell you who's there. Once you start looking into who is there, this is the same site. I'm sorry, it's a little light. So what you'll notice is that each circle represents 1 year of sampling, and each of those little crosses is one sample. And each little cross represents literally 1 teaspoon of soil that we've extracted all the DNA from. So it represents about 20 to 30 species of bacteria and as many fungi. And what you'll notice is there is a shift in these circles move around from year to year. And the reason why they move around is because who's there changed, but not how many changed. And that is very important because sometimes you'll see a shift in your community structure, but not in the numbers. And that is very important. Because sometimes who's there is more important than how many is there. And then we did something else, so we went back to our work pros and bench rows, and we saw exactly the same thing. 2 different sites, literally half a meter apart, very, very different communities present in those soils. That's the other take-home message. The soils are very fragmented in the Western Cape. So if you go to pristine fynbos, for instance, and I sample here, 1 meter away, I will have a completely different community. Most of those communities are dictated by the plants that grow there. So as soon as you take them out, you put a crop in there that's a monoculture, you tend to reduce that diversity very, very specific, and then you have this homogeneous group of microorganisms. Okay, so what we like to see in soils, something we call a resilient microbial community. Now, it doesn't matter what you do to them, some of them will stay because you'll have duplication of functions. So there's something we like to see in soils. So when you do this analysis, you look for things that can perform the same function. Now, that is something that's very easy to ask, very difficult to show. There's a lot of things that influence microbial communities. So now if you can imagine yourself what it is like in a little grain of sand, you have various organisms present, and then you have these various effects that influence whether they will be there, whether they'll grow, whether they'll produce all the nutrients or recycle the nutrients that you require them to do. And yeah, we've heard about no-tillage this morning. One of the things that I've read about in literature is that if you really want to disturb your soils and reduce your diversity, then you till your soils. That would really destroy those communities. And one of the first things that we did was to look at invasive species, and it's very similar to what you do when one plant species replaces another. So we looked into the riparian ecotones, which is just the river systems in the Western Cape, and I'll show you this as an example of how we can use DNA data to show how communities change and get a picture of what happens to microbial communities. And basically what my PhD student did was he sampled at rivers where there was no disturbance and rivers where there was disturbances from acacias, and then we went to sites where they removed the disturbance, or where they chopped out the acacias. And we looked at bacteria, and we found that they were very distinct, clear microbial communities. Once again, each circle represents a treatment, if you can think about it that way, and it seems to, well, at least for the bacteria at least, it looks like their presence is dictated by their water. So, terrestrial is down, forget it by Niki. Okay, so terrestrial down here looks very different to the wet bank, but that makes sense. You'll have different communities inside water than you would have on a terrestrial area. And then you have a nice transition zone, which is also, we were happy to see. But it looks like that water have a very, very, very big impact on which bacteria occurs. So we thought we've have a handle on things, and we did with the fungi, and if you notice there's no circles there. It's because fungi just occur wherever. They don't pay attention to water. So that was a little weird. So we thought, what do dictate their distribution? So if you do this kind of analysis, so just as it was very clearly explained to me, is all the red lines are fungi. So these are communities, and the blue ones are environmental conditions. And it seems that if the red line lies on the blue line, that is the factor that influences the distribution of that particular community. Turns out that pH, not so much, but all the other soil factors have a massive impact on how these communities are distributed. So you can manipulate your microbial communities just by manipulating your environmental factors. But then you need to know what you'd like to see in there. So we see that it's mainly influenced by invasion, in other words, the plant. So a fungus is very much influenced by the type of plant that occurs on that specific site. So we thought that was really interesting, and then we had another opportunity to test these hypotheses using an agricultural system. So because acacia tends to be a little bit difficult to test it on, we don't have control over the experiments because we cannot go and invade systems. And the other interesting thing that we saw before we go further is that once you remove it and you let fynbos comes back, you have your communities go back to an almost pre-invasion state. And that is a good thing to know, because you don't want to damage your soils irreparably. So in our search for looking for a crop system, so the question was, what happens if you have an indigenous plant that replaces all the other plants and starts becoming the crop? Would it behave in the same manner? So we sampled Rooibos and Honeybush teas from commercial farms as well as from wild stocks, and it was actually quite interesting to see. So once again, I'm just going to show you the Honeybush data just because, well, they all look more or less the same. Yes, my class does that sometimes as well. So what we find is that once again, if you look at diversity and we count the number of cells and bacteria species that we find in fungal species, there is no significant difference. This is where we come to the cool stuff that we can do with the DNA. But what we find is there's a very distinct shift from dry season to wet season. So, and that's another thing that you have to keep in mind, is that if you do a microbial analysis now, in 6 months' time, your community will be completely different. So they have these complete shifts in their community composition. They replace each other, and then they come back again. It's fascinating to observe. So when we look at this, we thought, what is the effect? So what is structuring these communities? We thought it was going to be the plants. It turns out it's not the plants. So it's not the farm practices, because the farms had different farming practices. It's not that. Turns out it is seasonal. So every time you go to a different crop, you go to a different system, it's something else that dictates how these communities perform, what they look like, and what the functions are. So what else can we do? So now we have a new technology. So I'm not sure how many of you are familiar with sequencing, but it's basically where we determine the number of, or the base pair sequence of an organism. And we can identify you based on your base, so your sequence. We used to do this one at a time, which is really time consuming, but now we have this fantastic new system where we can do parallel sequencing, which means we can do thousands at a time. And then we can generate things like this. So these are stacked charts, and each of those colors represents a bacterial group in the soil. So each of these bars is a soil sample, which taken at one of these farms, and each of these colors represents a bacterial group. So we can now get an indication of, in the soil sample, who are they really, so we can put names to them. And this is important if you want to know how many nitrogen fixers are there, how many organisms are there that can drive the carbon cycle. Those are really interesting, and I'm going to show you something that is, well, this picture is actually a little easier to see. So these are the community, these are the bacterial species that you'll find in these soil samples. So this is a commercial find, this is a natural soil. Looks very similar. That's fun to know. Well, it makes sense, because honeybush, when you plant it, you don't clear, you plant it in between all the other fynbos plants. It requires the bees that's there to pollinate, and it requires other organisms because it's really bad at getting its own nitrogen. Big difference between wet and dry seasons, and quite a big difference between the bulk soils and the rhizosphere. Makes sense. Your rhizosphere would be very different to your bulk soils. What was really interesting here is that as soon as you look at your whole community, remember, these are not things that we cultured on plates. These are, we just look at the DNA. You'll notice that there's these big light blue patches, and these are unclassified bacteria. In other words, these are things nobody has ever seen before. We don't know who they are. We don't know what they are, and we don't know what they look like. But it forms the bulk of our soil samples. So these things are doing something in the soil. We have no idea what they're doing. The other thing that's very interesting is the Acidobacteriaceae, these are the red ones. These ones, also quite a large part, and these are what we term unculturables. So there's 6 species known. We know they have quite a big genome, so they have all the genes that's required to produce a number of enzymes. We think they may be involved in carbon cycling and nitrogen cycling, but we don't know how to count them, and we don't know how to isolate them. So these are the things in your soils. So you should be aware of them. So how do we measure them? I don't know yet. So it's interesting. We see the same thing when we go into into the Rooibos. So it's a very similar picture when we go into Rooibos. Once again, we have a large number of bacteria that's unclassified. We can't tell you what it is. We can't culture them. Again, we see these acidobacteriaceae here. So this is these acidobacteria. What's very interesting, acidobacteria thrives in low pH soils, which is why we see them here. As soon as you put any form of agriculture, they tend to disappear. So the question is, what are they doing? And for me, it would be very interesting to know what these bacteria are actually doing in the soils and what can they contribute. And if they are important, what can we do in agriculture to stimulate their growth? Because you can't culture them in big tanks and spray them. You have to do something to your soils to try and encourage their growth. So this is some of the fun things. Now what we can also do is we can put these through a huge analysis and we can see if there are interactions between these bacteria. This is just a network analysis that one of my PhDs did. So each of these circles represents one of these bacterial groups, and the bigger the circle, the more the influence, the smaller the circle, the less influence it has. And if there's a line in between them, it means they interact. So this is wet season, not a lot of interactions, but as soon as you go into it was dry season, this is the wet season. No, it's the other way around. No, this is the dry season. Sorry, this is the right way around. You'll see a lot of interactions. So as soon as it becomes dry, these organisms clump together and they work together. But we're not sure how, and we don't know why, and we have no idea what drives this. So this is just the bacteria. And then we get to the fungi. Look at this. These are fungi. Millions and millions of them in soils. Many of them we can't see. Majority of them are saprophytic. So these are the ones that break stuff down. So if you have stubble, they would break it down and release nutrients into soil. Very few pathogens in natural soils, but as soon as you get into agricultural soils, you find your pathogen component increase. Then which brings us to a question. So at this particular point, I met somebody who sprayed Trichoderma species. I'm very interested in Trichoderma because it eats other fungi, and that's a fun thing to do. There's very few organisms that can actually eat other fungi, and if they can, they are of great economic importance. Now, my question to this particular farmer was, how do you know that it works? And he's like, it works. I said, but where did you get it? Oh, you bought it. I said, okay. What happens to the microbial communities that you spray this on? Because remember, we're now spraying a live organism in a community that lives like an organism. So you can think about this. If you take something in, that's a live organism, what would it do? So we were interested to know, can these commercial Trichoderma species replace native species? That's a very easy question. The next one was not so easy, as how many Trichodermas occur in South Africa? Which ones are native and which ones are introduced? Yeah, that was not it. So the answer was, I don't know. Nobody's looked at this before. So we went on a field trip, which is always fun to do, just because people here are interested in Trichoderma. It's a really old genus. People knew about it since 1794. We find it in soil, plants, but also in marine environments. So a number of them has been isolated from coral reefs, for instance. We like them because they are microparasites. In other words, they can eat a fungal pathogen, but they are indiscriminate microparasites, so they would eat everything else as well. They're not specific. In agriculture, they're used as biocontrol organisms, but they also produce metabolites, so they would produce indole acetic acids, which makes your plants grow better. But they can also make other fungi grow better, including plant pathogens. So think about that. So we have these crop growth promoters you can put in your seedlings, so one needs to think about using Trichoderma very carefully. Industry likes them because they produce enzymes. If you look at the genome of a Trichoderma, it has a huge genome, massive thing. Produces huge, huge numbers of enzymes, different kinds. So they're very important. So currently, we know there's 13 species in South Africa, only 13 species. The genus itself contains hundreds of species. 2 of them are commercial strains, they're used for biocontrol. The majority is unstudied. So we're importing an alien strain into South Africa, sprayed on our crops. The question is, what does it do to our native communities? Got to think about this. If I want to import a fungus from overseas, I need permits. Apparently you don't need it for Trichoderma because it's a crop, as a feed additive or an additive that you add. So, but anyway. What you also need to understand is that some of them can be human pathogens. So there's 3 human pathogens: Orientalis, Citroviridae, and Logibacchiatum. So 3 human pathogens. People that are at risk are people with immune deficiencies. Okay, so this is where we sampled. So we tried to sample as far as possible, as wide as possible. We isolated 178 Trichoderma strains, of which we put them in 11 groups. We did the full, complete taxonomy of these organisms, and we isolated a number of biocontrol agents. You will recognize the top one. So this is the one that you find in commercial applications, as well as Trichoderma harzianum. And then we also found 2 new species that's never been seen before. So they're novel to South Africa, they're indigenous to this country. So I have to ask the question, what do we do to these species when we spray aliens on them? So these are, just to give you an indication of the ones that we found. From this table, you can see how many of them has never been reported from South Africa. So these are new reports. They've never been seen here before. They are new reports to South Africa. So we do seem to have quite a bit of Trichoderma species in here. And if you don't know what it looks like under a microscope, I thought I included this picture. Okay, so this is what it looks like under a microscope. It's a really, really pretty little fungus, very delicate, but very effective in eating up other fungi. So now the question was, if you want to develop your own product, how well would our native species do if we compare it to the biocontrol agent? Then we did a little trial. So we used some vineyard pathogens, and these are 2 novel species that we described from South Africa. So only in South Africa they occur. They do very much a lot better than our control species. So these are the control organisms, the ones that we generally use as commercial sprays. Do very well. So we're very excited. And then we tried it on another organism, and didn't do so well. And then when we put it on botrytis, it seems to have helped some of the other pathogens, some of these botrytis pathogens. So some of these species can actually help other fungi to grow better, which is also interesting. So in a little trial that we do, we'll expect 10 next year, we took these 2 strains and we tested it against seedlings. And in our case, we used Honeybush and Rooibos because that's the current project we're working on. We made a mixture of rhizobia, which we isolated from the honeybush and the rooibos, and we put it onto little plants and we made them grow, and then we measured them. So what we have here is a little rooibos plant, a honeybush plant without anything, one with just rhizobium and one with trichoderma. So the trichoderma, we didn't put the fungus on, we just used the soup that comes off the fungus. So we just use the media it grows into, we spin out the fungus, we just use the swamp soap. And it has a lot of indole acetic acid in it and other growth promoters. So it seems to work very well. Didn't work so well for rooibos. And there's a number of reasons why we think it didn't work so well. So the take-home message is that if you have one species of trichoderma, it may work on one crop. And it's been said a couple of times that you need to find a strain that is crop-specific. And I think in South Africa we haven't clued up to that yet, and there's a lot of potential here to find things that are very regional specific and that can help us to increase yields and growth. So, if you don't remember anything else this afternoon, just remember soil is very complex. There is more under your feet than you can imagine. It is a living organism and should be treated as one. So you should keep it healthy as a whole, and not as one species. So agricultural practices that encourage and support diversity should be, we should try that out. I think it should work. So I need to thank a couple of people. I think I've got 4 minutes left. So these are the students that were involved in the projects, my collaborators from Stellenbosch, and then we had funding from a number of universities. And I'd be happy to answer any questions that you have.
Goed, terwyl ek vir Hoppies vra om gou nader te kom. Nogal een ding wat vir my interessant was uit Karin se praatjies en dis iets wat ons nogal moet mooi dink oor is oor al hierdie goed wat ons so spyt op ons goeders dat mense regtig moet dink voor mense net blindelings 'n ding doen. En hopelik kan van die werk wat ons nou begin doen het met van die goed wat die ouens by die boere by omkom, dat ons 'n toets en bietjie neutrale antwoorde kan kry, dalk kan dit bietjie meer inligting vir ons bring en om seker te wees dat dit wat ons doen werklik die moeite werd is.